Thank you for the invitation and introduction. It's my pleasure to be here. For those who are wondering, this is a DNA polymerase. This is RNA polymerase. I'll come back to this in a moment. So I will talk about the causes and the consequences of somatic structural variations in cancer. All right, so structural variations are also called genomic rearrangements. They are large-scale changes at the DNA, which often involves breakpoints, for example, deletion, duplication, inversion, translocation. Some can be fairly complex. And there are some overlap between SV and the copy number variations, which is CNVs, but there are also distinctions. For SVs, we focus on breakpoints. For CNVs, we focus on dosage change. For deletion, there's breakpoint, there's also dosage change. So deletion is both an SV and a CNV. And there are balanced inversion translocations. They have breakpoints, but no dosage change. So these are only SVs, but not CNVs. Then you have chromosome level aneuploidy, which means the dosage change of the entire chromosome. There are dosage change, but no breakpoints. So those are CNVs, but not SVs. So today I'm going to talk about three stories, two related to how they form in tumor, and the third one related to how they drive disease. Now, first, I want to give you an overview. What are we talking about SVs in a high-level view of cancer? So on the top, each dot represents a tumor and grouped by tumor types. And then on the left side, you can see sarcoma, ovarian cancer, breast cancer. They tend to have more SVs than the tumor types on the right. This is pediatric brain cancer, AML, thyroid cancer, which have relatively less. And then the bottom panel is showing you the relative composition of simple SV versus complex SVs. I'll come back to complex SVs in a second. You probably noticed that in each block, the left side samples tend to have more pink colors, which means the tumors with a lot of SVs in them are typically composed of a lot of complex SVs. And from this panel, you probably can appreciate that in every tumor type you look at, the spectrum of the number of SVs vary by 3 orders of magnitude for every tumor type. So there's a tremendous diversity. Typically, when you study a topic, you start with simple cases, then gradually you work on those complex cases. But in this case, we did it opposite. We started with the most extremely complicated cases, which is called chromophoresis. So this is one kidney cancer genome, and all these colorful lines represent individual SVs. The blue ones are intrachromosomal, the red ones are interchromosomal. You can probably tell that most of the chromosomes are relatively quiet, except chromosome 5 and 13. There are a lot of rearrangements going on between these two chromosomes. And the chromophoresis is discovered in 2011 by Peter Campbell group at the Sanger Institute. They propose this is a very bizarre event. It cannot occur as what we think the mutations usually occur in cancer, because the point mutations and other things, they accumulate one at a time over a very long period of time. And there's no mechanism known that can produce repeated translocations between two chromosomes. So this has to be a one-time event, which itself is also very bizarre because there's no known mechanism that can produce something like this as a one-time event. However, later on, there are multiple mechanisms have been shown that indeed this is the case. For example, micronuclei, during cell division, sometimes one or two chromosomes may be running at not as fast as the rest of the chromosomes. They are lagging behind. And after cell division, the chromosomes are in place and form new main nuclei, then this 1 or 2 chromosome will wrap into its own membrane called a micronuclei. And then in the micronuclei, the chromosomes actually will be broken down into hundreds of pieces by enzyme activities. And then some of these fragments will randomly stitch together into a new chromosome. And somehow that new chromosome would join back to the main nuclei, become a stable chromosome. And then given the enzyme activity that active along the entire chromosome, the breakpoints will be randomly distributed across the entire chromosome, depending on how many copies of the chromosome get involved and which fragment get retained, which fragment get lost. You would have this oscillating copy state between 2 or 3 state. And then sometimes one chromosome will have a break in the middle, and the cells do not like free DNA ends. So that broken end would tend to ligate to something else. And sometimes this would form a dicentric chromosome, which means one chromosome with 2 centromeres. And during cell division, that 2 centromeres sometimes can be pulled towards 2 different daughter cells. And even after the formation of 2 daughter cells, you can still visibly see a DNA fiber forming a bridge connecting 2 daughter cells. And then this bridge can be broken by mechanical forces. And some of these broken fragments can randomly, again, stitch together into a new chromosome. And oftentimes you would see the chromosome losing one side of the telomere because that's how it formed a dicentric chromosome in the first place. And sometimes this dicentric chromosome not necessarily become chromophysis, but the chromosome can break in the middle as a simple breaking end. And that is a raw material for the next cycle of called the bridge fusion breakage cycle. The broken end will fuse with another chromosome, form another dicentric chromosome, and then break again. Depending on where each break formed, then you would have multiple fragments getting gained or lost. So eventually you would have more copy states. And during these two processes, some of these small fragments sometimes can ligate head to tail together, form a circle. And this circle will eventually become circular extrachromosomal DNA and also called double minutes. So these fragments are very frequently found in tumor samples, and oftentimes there are oncogenes on them, so they are highly amplified. So in case you have never heard of chromophysis, one day I was watching a cartoon with my kids called Spies in Disguise. This guy was a spy. This kid was a scientist who made a potion supposed to turn human invisible. After the spy drinking that potion, his genome was undergoing chromophysis. I was like, wow, I was so impressed by Hollywood screenwriters. They are really up to date with science. So we use the PCOG consortium data, which involves over 2,000 whole genome sequenced tumors across 37 tumor types. And we use this algorithm called ShadowSeq to detect complex SVs. Basically, we're looking for a pattern like this, that you have a lot of SVs going on one or a few chromosomes, in opposed to these two cases. This one can be interpreted as individual SVs accumulating over time, but we are requiring that the breakpoints to cross each other because this is a pattern that unlikely to be explained by individual events accumulating over time. And this one, even though they cross each other, but it's all duplications, you can still interpret this as a repeated duplication accumulating over time. So this is a pattern that's unlikely to be individual events accumulating, but more likely to be a one-time complex event. So out of these 2,000 tumors, we found roughly half of the tumors carry some kind of complex event, and each tumor carry 1 to 2, occasionally 3 complex events. So then, since I already mentioned the different mechanism will produce event with different patterns. So with at least 2 or 3 known mechanisms, which one is more prevalent in tumor samples that we eventually see in the clinic. And even better, because the patterns somewhat reflect the mechanism, can we identify new patterns which may suggest new possible mechanisms? So some of you might have heard the mutation signature work from Ludmil Alexandrov using non-negative matrix factorization. Essentially, this is done in point mutation, but you can do the same for copy number variants, even for expression. That in point mutation space, that you have different molecular mechanisms generating individual mutations. For example, the cytosine deamination, which is spontaneous mutation, tobacco smoking, UV light, they all have different patterns. And for individual tumors, you have a mixture of different mutation signatures contributing to different relative abundance to individual tumors. Then it is mathematically feasible to decompose the mutation signatures if you have a lot of tumor genome sequencing data. And the non-negative matrix factorization is the algorithm that's been widely used for this purpose. So we tried NMF-based method for 2 years, trying to come up with ways to build input matrix and capture patterns and decompose signatures for chromosynthesis. But in the end, we realized it won't work, because for this method to work, you need a lot of these mutations accumulating in the tumor genomes. And you need them to be independent event accumulating over time, which is the opposite for chromosynthesis. Even though you have a lot of breakpoints within the event, but those are not independent event, and they do not accumulate over and over in tumor genomes. So what we end up doing is this algorithm called Starfish. We use 5 different features to describe the overall pattern of the event. For example, breakpoint dispersion score to measure the randomness of breakpoints across the chromosomes, and amount of copy loss, amount of copy gained, maximum copy number, and maximum amount of telomere loss, which trying to model that the chromatin bridge is starting with losing one side of the telomere. So then we did something simple. We assigned 5 scores to all the complex events we found in those tumors. And we asked one simple question: what if we put events that look similar together next to each other? And we performed unsupervised clustering. We come up with 6 clusters. And from now on, I will call these 6 different signatures. So we tried many different ways to construct clusters. We always end up with these 6 clusters, which means this is pretty robust. And then how do we know which signature represent what? We took advantage of a few published studies that they experimentally induce chromosynthesis by either inducing micronuclei or inducing chromatin bridge. So these are ground truths. And then we run our algorithm to predict what these events actually look like. So most of these micronuclei induced chromosynthesis being assigned with this greenish color, and most of these chromatin bridge induced chromosynthesis get assigned with orange color signature. So let's come back to this to see what is green, what is orange. Here you see that the signature 4, which is the green signature, has the lowest breakpoint dispersion score, meaning the breakpoints are evenly distributed across chromosomes. So that actually match what we know about the micronuclei. And then for the orange signature, signature 2, it has the largest amount of telomere loss, which matches what we know about the chromatin bridge. And then these are the 2 examples. This is the same kidney cancer genome I showed in the circle before, that now we are showing the chromosome 5 and 13 in a linear way, that you can still see the intrachromosomal, interchromosomal SVs, and the breakpoints largely evenly distributed across chromosomes, in opposed to this one, that the liver cancer genome, that breakpoints are relatively concentrated in certain regions of the genome and accompanied by losing one side of the telomere. And this is the sarcoma genome that several chromosomes get involved, and you see all these connections between them. And these, you see the very small fragments often get highly amplified to several dozen copies. So this is ecDNA, extra chromosome DNA. So now we have these 6 different signatures, at least 3 of them somewhat capture biological processes. And we have 3 new patterns or signatures, which may suggest they might represent new mechanisms. So one of these is a signature 6. I will point out the main property is that they have a highest breakpoint dispersion score, which means the breakpoints are highly localized, and they have a little bit of copy loss, but pretty much no copy gain. So this is an example of the prostate cancer chromosome that you have this very highly localized breakpoints, and every time you have a breakpoint, you have a little bit of copy loss, but rarely you see copy gain. So this is a signature we called hourglass chromothripsis. So now we can come back to this big question that out of all these known or new mechanisms, which one is more prevalent in tumor? Again, this is all the tumors summarized together. On this side, you can see adult brain cancer, sarcoma, esophageal cancer. Somewhere between 80% and 90% of the tumors have some kind of complex event. On this side, pediatric brain cancer, lymphoma, thyroid cancer, they have very few chromothripsis. And then you probably notice that prostate cancer here is very different from all others, that you have a big chunk of this new signature 6 hourglass. And then we have looked at all the mutations. We found this mutation of SPOP, which occur in 6% of the prostate cancer. They are correlated with 3 different signatures, number 6 hourglass, 9 micronuclei, and 3 large loss, which I didn't have time to describe. So at this point, we think SPOP mutation behaves somewhat like TP53. It probably does not drive the formation of individual event, but allows the genome to tolerate this kind of catastrophic events. So next, we turn our attention to simple SVs. What's the cause of simple SVs? Deletion, duplication, inversions. So in this case, we modeled thousands of tumor genomes. From the genomes, we predict a molecular mechanism. And from the mechanism, we predict 2 drugs. And based on one of the drug targets, and we kind of guessed what could be the drug mechanism. And based on the drug mechanism, we predict another dozen drugs. And we are able to experimentally validate all of these predictions. So this is motivated by the fact that BRCA deficiency, which is caused by mutations in both germline and somatic mutations in BRCA2, BRCA1, BRCA2 genes. These are the most well-known breast cancer susceptibility genes, and also involved in ovarian cancer, prostate cancer, pancreatic cancer. So the main consequence is they both are functioning in homologous recombination, and the mutation will lead to homologous recombination deficiency, which cause double-strand break repair deficiency. And then the consequences of genome instability is very unique. The BRCA1 mutations always lead to small tandem duplications. BRCA2 mutations always lead to small deletions, even though these 2 genes function in the same pathway. And for tumors carry BRCA1, BRCA2 variant, since they have homologous recombination deficiency, and this repair deficiency can be targeted by PARP inhibitors in the clinic. And individuals carry one of these variants may choose to surgically remove their breast or even ovary to avoid their risk of getting breast and ovarian cancer. And Angelina Jolie actually carry an ATM variant, germline ATM variant. It's in the same homologous recombination pathway. So the process we focus on is called transcription replication collision, as I showed in the title slide, that DNA polymerase, RNA polymerase, sometimes they crash into each other. And this collision actually occur in normal cells, but in a not so frequent. And in normal cells, there are repair machinery in place to fix the collision. And in tumor cells, as we know, pretty much every normal process can be somewhat compromised in one way or another, and sometimes being hijacked by tumor cells to be taken advantage of. So we are wondering, is it possible in a subset of tumors that this collision is not properly repaired? That means you would end up having SVs if the collision is the actual copy of DNA have to ligate with something in the genome. So how do we tell whether this is the case or not? So now this time we can use negative, non-negative matrix factorization to extract signatures, because simple SVs, deletion, duplication, inversion, they behave more like point mutations. They actually repeatedly occur in the genome that we can model the signature. This is done by postdoc Yang in the lab. So first we divide all the types of mutations, SVs, into four major types: deletion, tandem duplication, inversion, translocation. Then we break down into smaller size categories, because from the BRCA1, 2 cases, we know that size matters. Then we run this NMF-based signature decomposition. We are able to decompose in the same peacock cohort 13 different signatures. 4 of them are deletion signatures from small to large, 4 tandem duplication signatures from small to large, and a number of inversion translocation related signatures. So then, how do we know which signature might be the consequence of the collision, if there is any? So there's one nice property of this. So once you have this 2 polymerases collide to each other, that you have this half replicated fork, that this side is replicated, so there are 2 copies. This side is not replicated, there's only 1 copy. So then if some of these things would actually become SV, then you have a dosage imbalance. You have an extra copy here. And then you can also define replication orientation from sequencing data, that there's a RepliSeq method being developed that you can tell these sharp transition region, that the peaks represent early replicated regions, valleys represent late replicated regions, then the in-between sharp transition regions, you can define which is left replicated, which are right replicated. So then we overlay all the SV orientation with all the replication orientation, looking for potential dosage imbalance, that this replicated region is overrepresented in the SV breakpoints. So we did this in a peacock cohort over 2,000 primary tumors. We see TD3, TD4. These are large tandem duplications showing this dosage imbalance. And we can reproduce this in the Hardwick cancer cohort, which is all metastatic tumors, also more than 2,000 of them. So if this is really the large tandem duplication is the result of the collision, we would expect this to be dependent on gene expression. Then we classify genes into unexpressed versus expressed genes, and we do not observe any enrichment in unexpressed genes, but this enrichment is only show up in expressed genes. This is another level of evidence that the large tandem duplication actually captured the expected pattern of the result of collision. And we can reproduce in many different individuals. So this is a pan-cancer cohort. Then if we look at just breast cancer for a different cohort, and we use MCF7 to infer replication orientation, this is a breast cancer cell line. We can reproduce the signal. And if we only focus on liver cancer, use a liver cancer cell line to define replication orientation, we can still see this expression-dependent dosage imbalance. And then I show you another overview that how often do we see large tandem duplications in different tumor types. This is the peacock cohort. You can see that on this side, uterus cancer, ovarian cancer, stomach cancer, they tend to have more large tandem duplications. And this is the metastatic tumor cohort. You see the ovarian cancer, esophageal cancer, again, on top 3. And now I want to point you to this prostate cancer that there's a small group of tumors seems to have a lot of large tandem duplications. This is because in metastasis prostate cancer, this is involved with CDK12 mutations. This is also true for ovarian cancer, that CDK12 mutations are correlated with large tandem duplications. And CDK12 mutations are more frequently found in metastatic prostate cancer, about 10%, but in primary prostate cancer, it's about 1%. And in metastatic prostate cancer, the mutations are correlated with the worst outcome. So now if I conclude that CDK12 mutations cause transcription replication collision and cause large tandem duplications, you probably don't believe me. I don't believe that either. So we collaborate with Jonathan Cho at UCSF. So we performed CRISPR knockout in 3 different cell lines, 22RV1, C42B. These are prostate cancer cell lines. And the PNT2 is a normal prostate epithelial cell line. So in all 3 cell lines, we perform biallelic knockout. You can see that CDK12 proteins are completely gone. And then, I know most of people here are bioinformaticians, but bear with me for a second. So this is the R-loops. It's the DNA RNA hybrid. So in the control cells, we have a mild baseline level of R-loops. And then in the CDK12 knockout, we have increased amount of R-loops. And this can be rescued by adding wild-type CDK12 proteins. And then we can use RNase H to digest the R-loops to bring down to the baseline level. And then with, again, in the control setting baseline, you have minimum level of transcription replication collision. If you knock out CDK12, you have increased level of collision. And if you add CDK12 wild-type protein, you can rescue this phenotype. And if you add RNase H, to remove R-loops, this can bring down the collision level back to the baseline. So in the end, before we talk about the model, we also performed a very complicated single clonal expansion experiment. So initially, when we performed CDK12 knockout, we pick individual cells, expand into clones, so make sure that all the cells are homogeneous, carry the same CDK12 mutation, and confirm it's a biallelic clean knockout. And then we grow these cells for an entire year, allowing the cells to accumulate individual variants for long enough time. And then after 1 year, we again select individual single cells, expand them into clones, and we do this for the control cells again. Then we sequence all these expanded clones compared to the parental line, looking for somatic variants accumulate over the period of 1 year. This second round of single cell expansion is necessary because presumably these cells will accumulate alterations independently. So every cell is different. We really have to pick single cell and expand again. So then in the control cells, we have some variable number of somatic SVs. And in the knockout cells, it's very hard to see from here, but if you just look at the fraction of large tandem duplications, you can see the difference. In the control cells, about 5% of the SVs are large tandem duplications, but in the knockout cells, you have 15%, which is much higher. And this control clone looks to be so much higher than everything else. This is because this clone actually acquired another mutation in DNA repair pathway. So just from this experiment, we can conclude that loss of CDK12 is indeed promote the large tandem duplication formation. So now we can come to this model that we think what's going on. The normal function of CDK12 is to phosphorylate RNA polymerase 2. With biallelic loss of CDK12, this RNA polymerase phosphorylation will be deficient. And the phosphorylation is known to be playing important roles in transcription termination, which means once you finish transcription, the polymerase is supposed to be unloaded from the DNA template, and the newly synthesized RNA should be unloaded. And now the polymerase somehow gets stuck on the DNA because of this deficient phosphorylation that will lead to the newly synthesized RNA to be stuck on the DNA template. And we know that if you digest the R loops, you actually can bring down the collision back to the baseline level, which means the polymerase itself isn't the problem. The problem is the RNA gets stuck on the DNA, which become essentially a roadblock for DNA polymerase. And once the DNA polymerase come in, run into R loops and crash, and in normal cells, this would be properly repaired. And even in the CDK12 knockout cells, most of these events are still being properly repaired. And even after growing the cells for a year, we have accumulated like a dozen large tandem duplications, which means 1 per month. So still not very frequent. So what happened after that isn't very clear, but based on the fact that we get large tandem duplication, we think this polymerase would somehow roll backwards hundreds of kb upstream and restart replication and go through the same stretch DNA twice. That's how you get tandem duplication. So if the formation of large tandem duplication represents some kind of repair deficiency, can we drug this deficiency? And we took advantage of this PRISM drug screening data, which screened over 1,000 compounds in about 500 cancer cell lines. So we asked a simple question. For those cell lines carry a lot of large tandem duplications, what kind of a drug are they specifically being sensitive to? And we found 2 drugs, a V1 inhibitor and a PARP inhibitor. And we are able to again, experimentally validate this in multiple V1 inhibitors we tried, and we are able to kill CDK12 mutant cells much better than the wild type cells. Wild type cells are essentially resistant to all these drugs. And then the V1 inhibitor work is because that V1 is in the G2M checkpoint pathway. ATR is the most upstream protein that detects DNA damage in S phase. Then if damage is detected, ATR would phosphorylate check1 phosphorylate V1 phosphorylate CDK1. And this pathway is in place to make sure that DNA replication is complete before cells can enter mitosis. And since the cells with CDK12 mutations, they have trouble replicating DNA. If we apply V1 inhibitor as well as any of these inhibitors that are blocking ATR or check1, that you block any part of this pathway that would allow the cells to enter mitosis prematurely, that will lead to cell death. So we think this is a very exciting study. We submitted a paper to Nature Cancer, got reviewed. All 3 reviewers are pretty positive, but asked 50 experiments to be done before publication. We spent a year, did all those experiments, and the second round review, 2 reviewers says everything is good. We are very happy with what's being presented here. Reviewer 3 said, this study lacks novelty and scientific rigor for any major journal. And the editor emailed me the review before making decisions saying, hey, this is the review comments. What do you want to do? We had an hour-long Zoom conversation with the editor explaining the novelty, explaining how we design all the experiment with rigor. And editor said, why don't you write down everything you said in a document and we'll discuss in the office? And then we sent out the document. It went back to the same reviewer. 2 months later, the reviewer said, I'll hold on to my previous opinion. I was about to freak out. Editor accepted the paper. So yeah, that's fortunate. So last, I want to tell you how do these SVs change 3D genome structure, then promote disease. So there are many different potential ways of SV can drive cancer. You can delete a tumor suppressor, or you can duplicate the oncogenes, or you can have translocations or other types of events from gene fusions. This is the Philadelphia chromosome balance the translocation between chromosome 9 and 22, which is very frequent in chronic myeloid leukemia. It occurs in 95% of the CML patients. And there are drugs targeting this fusion proteins because the fusion proteins are actually one of the best drug target for multiple reasons. One is they don't exist in normal cells. And tumor cells, the proliferation solely depend on the fusion proteins. So if you can screen for small molecules that with low toxicity to normal cells, and at the same time, you can block the function of this fusion protein and you have a drug. In fact, the first drug targeting this BCR-ABL fusion is one of the first target therapy drug in cancer. So then back in the day, we studied this rare type of tumor, chromophore renal cell carcinoma. We noticed that about 12% of the tumors have SV breakpoint around the TERT, never inside TERT, always around TERT. Whenever you have breakpoint around TERT, you always get activation of TERT. And this is a phenomenon is already known for decades called enhancer hijacking, that protocol oncogene that acquire an enhancer from other places of the genome. And this one is actually this same tumor I showed you earlier over and over, that TERT is right here on the tip of chromosome 5. It actually acquired an active enhancer from chromosome 13. And TERT is a telomere reverse transcriptase, which is required for cells to become immortal, that you need the telomere to be there for the cells to divide nonstop. So then we developed an algorithm called Hyena. Essentially, we use all the tumor genome transcriptome sequencing data to predict candidate enhancer hijacking oncogenes. So it's pretty complicated, but we want to use somatic SVs we detect from whole genome data and gene expression measured from RNA-seq. We want to predict candidate oncogenes that activated by enhancer hijacking. The key step is here. We do a normal score regression, which is necessary because many of you probably know that Jessica Lee has a paper, a model gene expression in tumor that it's so heterogeneous. However, you split samples randomly into two groups and you run differential expression analysis, you always have hundreds of genes to be actually differentially expressed, which doesn't make sense because those are randomly selected groups. That is because the expression is so heterogeneous in cancer that it's really hard to model. The best way to model is to use non-parametric ways. So the normal score is our way of modeling expression non-parametrically. So first we rank gene expression from all the tumors. Then we map the rank to a normal distribution. Then those z-scores are the normal score we use to do regression. And you can do all kinds of model selection in regression. And also this score is based on rank. So the outlier doesn't affect the outcome too much. And we also model a lot of confounding factors such as tumor purity, copy number, age, sex, and other principal components that's being used to explain some unobserved variation. And then essentially we're looking for SV, repeated SV around the gene that correlated with a high gene expression. So we did that in over 1,000 tumors, again, in the PCOG cohort, because only half of the PCOG samples have RNA-seq. Then we found the pancreatic cancer has the most candidate genes, 19 of them. And if you look at what type of gene are they, half of these genes are protein coding genes. That means the other half are all non-coding genes. Then in pancreatic cancer, we focus on this link RNA, top1 AS1. There are SVs affecting this gene in about 10% of the tumors. And if you look at tumors without SV, you see a significant change in expression, even though the fold change is only 1.7, not that dramatic, but we still decide to focus on this gene more. And then we looked at several dozens of pancreatic cancer cell lines. We picked 4 cell lines. 2 of them have high expression of this gene. 2 of them have low expression of this gene. And I want to point out this cell line, PA2 8988S, and this one, 8988T, they come from the same patient. One has high expression, one has low expression. And this cell line is known to form metastatic tumor in mouse. This one is known to not to form metastasis tumor. And then we collaborate with Francois Spitz at Human Genetics in our university, did a high-C experiment on all these 4 cell lines. And so for high-C, we are essentially mapping 3D genome interactions. And when you draw this in a linear way that you are showing, I'm showing the top 1 AS1 region, this is the location of this gene, and you see these off-diagonal hotspots. These represent somatic SVs that exist in these samples. And once you zoom in, you don't see much somatic SVs around. These ones are all too far away that to form actually new enhancer promoter interaction. And then in these 2 cell lines coming from the same patient, we did a TAC-seq showing that the promoter of top1 AS1 is more open in this highly expressed cell line, but less so in the low expressed cell line. So we think this cell line have high expression is due to transcription regulation. And then in this case, this is a high expression cell line, this is a low expression cell line. We see that you see there's a very not so obvious new domain forming around top1 AS1. And we went through a lot of trouble, eventually resolved what happens. This cell line has 1 copy of a wild type chromosome, and the top1 AS1 is the blue line here. And then this thing would duplicate again and self-ligate into an event called fold-back inversion. And this chromosome, there are 2 copies of that chromosome, and this part would translocate to a different chromosome. So then all these together form this kind of subdomain that you have 2 top1 AS1 gene within this domain. So since we are looking for oncogene, does this gene actually promote cancer? A graduate student, Anqi Yu, did a lot of experiments, just summarize here. When you overexpress this gene, that would lead to elevated cell invasion and more tumor metastasis. And so this is done during COVID. And in COVID, most labs would shift from wet lab to dry lab. We did the opposite. We shift from dry lab to wet lab. And I knew if we only do 1 cell line, it's not enough. We did 2 cell lines. And I knew if we only do cell culture experiment, it won't be enough. We did a mouse experiment. And then after review, reviewer number 3 said, I don't know much about algorithm, but what I know is your experiment is too problematic. You only did 2 cell lines, which is not enough. I want to see 4 cell lines. You only did overexpression, which is not enough. I want to see knockout. You only did tail vein injection. It's not enough. I want to see orthotopic injection. I'm a bioinformatician. I had to Google what exactly is orthotopic injection. which means you inject the tumor not under the skin, or in the tail, but you inject to the relevant organ of the disease. In this case, it's pancreatic cancer, then you have to inject the tumor cells into pancreas. And we did all that. This is the knockdown. Even by knocking down this top one AS1 gene by 50%, we can compromise the ability of these two high expression cell lines that you can reduce, you can pretty much eliminate the ability of cell invasion. And this is the orthotopic injection that you not only increase primary tumor, you can also promote the spleen metastasis. So then I want to point out this again, this 85, 88, 89, 88S and the other cell line we used earlier, this one, they come from the same patient. And there are a lot of difference in the genomes. One have aneuploidy, the other one doesn't. But just by manipulating the expression of this one gene, we can completely flip the phenotype. So we think the function of this linker RNA is pretty robust. So to summarize, we have an algorithm called Starfish that we can infer there are 6 different complex SV signatures, which may represent 6 potential molecular mechanisms. And we've shown large tandem duplications are driven by transcription replication collisions, and the CD12 mutant tumors are sensitive to multiple G2M checkpoint inhibitors. And our algorithm Hyena can detect candidate oncogenes activated by distal enhancers. With that, I would like to thank you for your attention. I believe I've mentioned all the people in the lab. I'm happy to take questions.